So, let us get started. So, what I thought I will do for today is start talking a little bit with the biology and leave the physics from next week onwards, just to get used to some of the terminology that we will be coming across again and again in the course. Okay. Here is a DNA double helix, right. So, this at least all of us know, the DNA double helix contains our genetic information, the genetic sequence, and what this DNA double helix is composed of is these 4 compounds: adenine, thymine, guanine and cytosine, A, T, G and C. These make up the genetic alphabet, and then if I have this DNA double strand, something like this, if there is an A over here on one strand, there is a T over there on the other strand. Okay. If there is a G on this strand, there is a C on this strand, and so on. So, these 2 strands are complementary. So, on one strand, if you have something like A, T, 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 A, G, T, T, on the other strand you would have T, C, A, so on. Okay. So, these are called this A, T, G and C. These are called the bases, and then this pair in the 2 strands of the double helix is what is called as a base pair. And then you have 2 things. So, when I have a DNA double helix over here, from a physics point of view, or from I have 2 sort of things that is going on over here. I have a string of these letters, okay, of these 4 letters A, T, G and C. So, there is a sequence information. So, there is a sequence, right. So, when I say I have I have a genetic sequence, it means that what I have essentially is something, something like that. Okay. So, here is my DNA sequence, and that is one representation or one way of understanding the hereditary information in our cell, right. So, the sequence information, but that is not the only thing. The other thing that is there is the physical vessel that is carrying this sequence, right, and which is this DNA double helix. So, there is a polymer as well, right. So, there is this polymer as well, on which is comprised of all of these bases, right, and that is a physical object, right. So, it's like a string essentially, and being a physical object, it has physical properties, right, and those physical properties have consequences. So, the sequence has consequences. The physical properties of the polymer itself also has consequences. Okay. So, depending on what you are interested in, you might want to focus on one or the other of these aspects. If you are a geneticist, you might want to focus on the sequence, mutations in the sequence, what functional changes do mutations cause. If you are maybe a physicist, you might want to focus on this polymer, what is the structure of the polymer, how can I change the structure, and so on and so forth. Okay. Why is this important? This is important in some sense, because if you think about it, all the cells in our body have the same genetic sequence. Okay. So, before I say that, any idea of, if I take the human sequence, human genetic sequence, how many base pairs are there in the human genome? Order of magnitude estimate? Anyone had? So, this is of the order of 10 to the power of 9 base pairs. Okay. So, I think roughly about 6 point something into 10 to the power of 9 base pairs. Okay. So, it's a string of letters, a hugely long string of letters. Okay. So, there is massive amounts of information that is over there, and you can imagine that. But this 6 into 10 to the power of 9 billion base pairs that is there in every cell of your body, right. But all cells, of course, functionally are not the same, right. You have your whatever heart cell, you have your skin cell, you have your liver cell, and so on. All of them have exactly the same sequence, right, because that is your genetic code, that's your genetic signature. So, something else is going on, which tells one cell that, hey, you go and be a heart cell, which tells another cell that, hey, you go and be a skin cell, and so on and so forth. Okay. So, what differences could there be that could lead to this sort of differential types of cells, differential cell types? Any ideas? Arrangement of DNA, arrangement, orientation, and foldings. What do you mean folding? Excellent. So, this is an extremely important aspect in some sense. Anything else? Anything else?
Cell types will get differentiated based on the requirement.
Based on the requirement, but of course, but that has to do with different proteins getting expressed in different cells. So, differential expression of proteins, it's related to that, but this is actually a very interesting, this is actually a very important point, the first one that you brought across. You had some question? Oh, it's it, I see. Because you see that if you take this billion base pair long string, you could ask that how long is this string, right? And each base pair is roughly about 3 base pairs in a nanometer. So, each base pair is roughly about 0.33 nanometers. Okay. So, if you were to take this string and you were to stretch out the string, roughly you would get about 2 meters, right, 6 into 0.3. So, the string is something like 2 meters, and you have to put it inside a nucleus which is about of the order. So, this string stays inside a nucleus which is of the order of some few microns, okay. So, which is 10 to the power of minus 6 meters. So, it is good to have an idea of the numbers and scales that are involved. So, you take this 2 meter long string and you package it inside an object which is about 10 to the power of minus 6 meters. Okay. And the way you do it and the way the cell does it differs from cell type to cell type. So, there are organizational, like he says, there are folding principles that are involved, and that in some sense is tied to what is the function of each cell. Okay. So, there is the sequencing, there is the sequence aspect, of course, which is this whole string of A, T, C's and G's, but there is also this folding aspect which says that if I take this DNA double helix, how do I fold it inside this nucleus, which is of the order of microns. Okay. Since we are talking a little bit of biology today, the way this DNA folds is a hierarchical process, which means that it goes through various multiple steps. So, one of the mostly it is unknown. So, we do not really know how it folds. Okay. We know a little bit, and what we know is that if you take this DNA double helix, what it does is that there are these proteins called histone proteins, okay, and this DNA folds around these histone proteins to form this object which is called as the nucleosome, and then there are many such nucleosomes. So, this is this whole DNA, and this nucleosomes sort of further fold on themselves to form what is called as a chromatin polymer, and then this chromatin somehow folds and forms chromosomes which are then folded inside the nucleus. Note that I get vaguer as I go up scales. That is because we do not really know what is going on, how it folds and so on. We know some parts, but we don't know the exact mechanisms and the exact details. And another way, for example, that cells sort of say that what is there apart from this genetic information is modifications of these histone proteins. So, you have these histone proteins. These histone proteins have charges, okay, and by playing around, the cell can play around with these charges through some chemical reactions, which are called post-translational modifications. Let's not get into some chemistry. Let's not get into that. But it can by playing around with the charges, you can understand that it can affect the folding, because if you have some polymer which is, let's say, positive charges, negative charges, versus if you have some polymer which is positive, negative, just to give a random example, how this polymer will organize differs from how this polymer will organize, because the physical forces play a role in the organization, right. So, therefore, if you have different charge distributions, you will have different sort of organizations, and so the way the cell does that is to have some post-translational modifications of these histone proteins. Now, what is the role classically, what is the role of the genetic sequence? The genetic sequence produces codes for proteins, right? Do all of you know how it codes for proteins? How do you know, all of you know what is a gene? Anyone? What is a gene? You all heard of a gene, right? Sibgat, what is a gene? Genetic material, but my genetic material is this string of letters A, T, C, C, G and so on, right. So, what a gene is, so I have this sequence A, T, C, C, whatever, this billion base pair long sequence, right. What this sequence does is that the main function of this sequence is to produce proteins, okay. And the proteins then go and perform various functions which allow the cell to live, right. So, the cell lives because proteins are correctly produced in correct amounts at the correct time. How does this how do I go from this sequence into these molecules which are into these protein molecules? The way one does that is to say that is through a process which is called transcription and translation. So, there are regions of this gene, there are regions of this whole genome, which codes for certain proteins, okay, through an intermediate which are called RNAs, which then produce proteins. Let me see. What is a protein? Before I say how it goes from here to here, first let me say what is a protein. What is a protein? So, this is my DNA polymer, which has this sequence, which is made up of 4 letters. This is an alphabet comprised of 4 letters A, T, C and G. This has this sequence information and it has this polymer structure like we discussed, okay. So, that is my DNA. The output is the protein, and what is a protein? A protein is something similar, except that the alphabet for proteins consists of 20 letters instead of 4 over here. The alphabet for proteins consists of 20 letters which are called amino acids. So, there are 20 amino acids, okay. And these amino acids, so let us say amino acid 1. So, you can now think of a sequence just like you had a sequence of letters over here, right, in the DNA. You can have a sequence of amino acids, amino acid 2 and then maybe again amino acid 1, then maybe amino acid 20 and so on. So, you now have another string of letters, except that this string of letters are amino acids, okay. And this also, so this has this sequence information and this also has some sort of a structure, okay. So, what are proteins? Proteins are sequences of amino acids that folds into certain form and performs therefore certain specified functions. Okay. All right. So, now let me ask a question. I will tell you. Before I tell you, let me ask a probability question. Okay. So, here I have a string of letters which is made up string of whatever word or book or whatever, which is made up of 4 letters A, T, C and G. Out of this, I want to code for 20 amino acids, okay. So, what is the minimum length of, so I can think that for example, I don't know, if I have a sequence over there which is A, G, T, E, A, C, something like this, right. This if the cell sees this sequence, it produces a protein, it produces an amino acid 1, some particular amino acid, okay. If it sees a different sequence, it produces amino acid 2 and so on, okay. Now, what I want to ask is that what is the minimum length of this sequence such that you can code for 20 amino acids, just pure elementary probability. Is the question clear? So, there is some code. So, this is the genetic code. So, and what I want to ask is that what is the minimum size. So, it's if each unit of this code is what I call as a codon, what is the minimum length of a codon such that I can code for 20 amino acids. 10 to the power of 9 divided by 20, okay. Why? Explain. 20?
Sir, amino acids are different.
There are 20 different amino acids, just like there are 4 different letters over here, there are 20 different amino acids over here. Okay. Now, somehow I want to go from this sequence to this sequence, and I want to ask what is the minimum length of these individual codons that I need to have in order to code for 20 amino acids. 4, okay. Why? So, explain the factorial 4 once. You can have repeats, right? Of course, like for example, I don't know. I mean, here I have a T followed by a T. Wait, earlier you had 4, now you have 4 raised to 4. It's a very big difference. So, what is the minimum length of the sequence? 4 raised to the power of 4? 3. Why 3? Yes.
You can make this 16 possibilities out of length 2.
Correct.
So if we extend to one more, 64.
Correct. So that is indeed correct. So the minimum length of this these codons that you need is 3, right? Because in the first position you can have 4 possibilities A, T, C, and G. In the second position you can have another 4 possibilities. So if you had codons of length 2, you would have 16. You could code for 16 possible amino acids. If you add a codon, another codons, if you have codons of length 3, then you could code for in principle 64 different amino acids. And so that's in some sense the minimal length that you require in order to code for at least 20 amino acids. And it turns out that, you know, in biology the answer is indeed 3. So a codon is 3 nucleotides, okay, or 3 bases. So you have this genetic code, you read this in groups of 3, and each group of 3 codes for some specific amino acid. Okay. So if you read CTC, that codes for some amino acid. So let me see, I think I have the table over here somewhere, over here. I will go back to that animation. Okay. These are your 20 amino acids, glutamic acid, aspartic acid, alanine, valine, and so on and so forth. Okay. So there are 20 amino acids that you have. And here are your sets of triplets. So because you have 64 possible codons and 20 amino acids that you have, there is some redundancy built in, right? You can have many codons that code for the same amino acid. So for example, if I read G and then A and then G, so GAG, this sequence codes for this amino acid glutamic acid, but so does GAA, right? So GAA also codes for this amino acid, the same amino acid, glutamic acid. And there are various levels of degeneracy. For example, valine, I can see this amino acid valine has 1, 2, 3, 4 codons that code for the same amino acid. Some might have, I don't know if there is anything apart from the start codon, which is completely non-degenerate. Yeah, I think that's the only one. So there is this special amino acid over here, which is called methionine, MET over here, which is AUG, right? By the way, what is U? I was talking about 4 letters, A, T, C, and G. This picture does not have a T, but it has a U substituted for it, okay? Yes?
So U is uracil, it is not present in DNA, it is found in RNA instead of thymine.
Correct. So when you go from, in which I still have not said, when you go from this DNA to this RNA, all the T's get replaced by U's. So, you know, if the sequence over here is CTCG, in the RNA the sequence is going to be CUCG, okay? And then you will read it in groups of 3. So that's why in this table over here, you have U's instead of T's. But so what I was saying is that there is this special amino acid over here, methionine, which is AUG, which is called as the start codon. So AUG, which is called as the start codon. What does it mean? What it means is that I have this 10 to the power of 9 letters, right, over here. not all of them have a functional role in protein production. Okay, they have other roles, but not all of them have a role in this amino acid production or this protein production. Okay, this regions of the genome that actually code for amino acids are what are called as genes. Okay. And the rest of the genome does not produce amino acids. So, a gene is in some sense therefore, the functional, or at least from this perspective, the functional segment of the DNA. When you have a gene, you read your nucleotides in groups of 3 to produce these amino acids. Okay. Now, where do you know where does a gene start? A gene starts wherever you have this sequence AUG, okay, which means that if I have this, so therefore this,
Sir, ATG in that sense.
in here you have AT, so therefore maybe let me draw something like this. So, I have this sequence, I start reading, I start reading, I start reading. Whenever I see an ATG, that means here is where a gene is starting, okay, and from here is where I will start producing these amino acids.
Sir, can we count this backwards like
Huh? Over here?
Yes, like GTA is written, will it become?
So, that is called as these reading frames. So, in principle, yes, you can have different reading frames. So, for example, even if I forget reading backward for the time being, let me just consider one strand of any one strand of this double stranded DNA. Maybe you have started reading ATC, CCT. So, maybe you have started reading from here, right? This is your first thing. So, you have read ATC, CCT, then CGA, then TGA. So, you read in groups of 3, you have passed this sequence, but you have not actually passed the methionine ATG sequence. Okay. So, therefore, it is still not in that reading frame, there is still that start codon has not appeared. So, it depends on how you read the genetic sequence, and in principle it is also possible to read from the other end. I actually have an assignment question on reading frames later on. So, so you have this special codon, which is the start codon, which tells you AUG or ATG on the DNA double strand, which tells you that when your actual genes are starting, your genetic gene is starting, and then you start reading from there and start producing amino acids. Okay. So, what is the role of this RNA in the middle over here? So, what this RNA does, so this is a multi-step process. So, what this RNA does is that this RNA is another polymer. Okay. So, it's another sequence like this. It is exactly the sequence of the DNA. Okay. It's a single strand instead of the double strand, but it's exactly like this, except this nucleotide thymine is replaced with this new one, which is called uracil. Okay. So, it's if you were to produce an RNA over here, it would read something like A, U, C, 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 T, C, and so on, except that this RNA are shorter fragments, in that you start producing RNA from this start codon, and you produce it until you reach a stop codon, and the stop codon are things over here, UAA, UAG, and then UGA. Okay. So, there are these three sequences that tell you when to stop, that tells you when the gene has stopped. So, you have this protein that is called an RNA polymerase that comes, it slides along the DNA, okay, this hugely long polymer. When it sees that this gene has started, it then starts producing copies, okay, and maybe this, I do not know. So, it produces and it starts producing until it reads one of the stop codons. So, what you get is a smaller fragment, okay, which has this gene region that has been transcribed into this polymer. Okay. And then from here, these are, well, there is more biology, there is splicing, and there is introns and exons, and so on. But essentially, if I consider it, what it then does is that it then takes the sequence in groups of 3 and produces amino acids, and you finally get a protein. You get a sequence of amino acids, which then folds into a protein. Is this sort of clear? The DNA is continuing. The DNA is a polymer, it has this whole sequence. Only the sequence which correspond, only the regions which correspond to genes produce proteins. Okay. And only those regions get transcribed into RNAs and then translated into proteins. So, this is called, this process from DNA to RNA is called transcription. And from RNA to proteins, amino acids is called translation. No, so I mean, if you are asking that there is there a chunk of sequence that then repeats again and again and again? No. Yeah, I mean, so what, it's a difficult thing to do, I mean, in that sense. So, what people generally focus on is on studying only the gene regions, okay, the regions that actually code for amino acids. You could ask that, well, how much of this billion base pairs, what fraction of these are actually genes? So, what fraction actually codes for proteins? Any idea? For example, well, it differs from organism to organism, but in humans, any idea? Humans, I think, is closer to 2% roughly. Okay. So, the sequence that actually codes for proteins is of the order of 10 to the power of 7 base pairs, which is still quite a bit, but at least it is not billion. So, there are various interesting questions that come up. So, for example, you could ask that what happens to the remaining 98% of DNA, right. So, that has a role in regulating. So, for example, this region codes for some particular protein, right. So, you have, here is where a gene has started, you have produced an RNA from there, you have produced a protein from there, okay. So, you have produced some protein. Does that mean that the cell is continuously producing this protein? No, right. The cell produces a particular protein when it is needed, okay. But the sequence is always there, because the sequence is the sequence, there is nothing you can do about your own genetic sequence. So, what these other, so earlier these used to be called, what was the name? So, earlier when people did not completely understand the role of this, even now we do not understand the full role, but earlier people used to call it junk DNA, now we do not call it that. We call the gene part as coding regions and the other part as non-coding regions. And the non-coding regions have a role to play in saying that when will a protein be expressed. So, it has regulatory roles, for example. So, it has a regulatory role.
What if there are multiple start or stop sets of these?
So, generally if you have a start codon, you continue reading until you have reached one of the stops. So, that signifies the end of one gene. So, that would be gene 1.
If there is a start in between a start and a stop?
Is there? I do not know actually. It is a good question, I do not know. Is there? I will ask the resident biologist. Yeah, maybe it is there, and then just this methionine just gets treated as an ordinary amino acid in the sequence. Not sure, I will check and let you. Right. So, what was I saying? Okay. So, now there is a nice video somewhere in one of these slides which explains.
The DNA double helix contains 2 linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out.
So, here what you have is a protein, this blue thing over here is this RNA polymerase protein. What it does is that it binds onto DNA, right. It slides along this DNA, and then when it reaches the start codon, it starts producing this mRNA polymer, and it will keep on producing this mRNA until it sees a stop codon.
Top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form. Spliceosome then brings one end of the intron is cut, disassembles. It's only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer.
There was one level of copying that happened over here when you went from this DNA to the RNA. There is another level of copying that is happening over here inside this ribosome, where again you are reading now these sequences in threes and producing the particular amino acids. If you read one particular amino acid, one particular sequence, the corresponding amino acid gets added on, and then you get this protein that this red thing is the protein strand that is coming out.
Molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message.
So that's the central thing. So you have the DNA. The DNA produces RNA, then this RNA produces proteins. This is called the central dogma of biology. You're eating protein. What you're getting is, what your body is using is not those actual proteins, but these amino acids. So it's broken down into amino acids. These amino acids are the raw ingredients which the cell then uses to make whatever particular protein that you want. So you're not eating the proteins directly in some sense, okay? But you're getting the raw materials for the production of proteins because it's the same amino acids that are involved. So this is a little bit of biology. Having done a little bit of biology, so this is the central dogma and so on. So now you could ask that, well, like I said, so here I have this protein production. I have my genetic sequence. My genetic sequence is read to produce amino acids, and these amino acids fold to form proteins. Is this protein production happening continuously, right? So if I look at the number of proteins as a function of time, it could be, in principle, if I say number of proteins as a function of time, it could be something like this. I always have my genetic sequence. If the genetic sequence is always being read to produce proteins, I would have, in principle, I suppose, a growing number. Okay. So if I say that here is my number of proteins as a function of time, right? I could say that, well, there is some bad symbol. So let me say there is some birth rate, right? Because you always have your DNA, you're constantly producing proteins. You are going to grow the number of proteins with some constant rate. Okay. How could I get to something? Maybe that seems bad. If I kept on producing proteins as long as the cell was alive, my cell would soon burst, right? So what could be a way to sort of taper down that number? What could I say? I could say that, well, proteins have a finite lifetime, in that after some time these protein will degrade. Okay. So I could say that, well, I will have some death process, right? Some death process. Something like that, and maybe in the steady state, then it will go to something like this, right? And that would be my number of proteins over here. That would be my number of protein molecules as a function of time. So that would be nice in some sense. You could then ask me that, well, how many proteins of this particular type do you have in this cell? I could look at this curve and I say, well, I have 200 proteins. I neglect this initial transients, which in physics we always do. We say that, well, as long as you have reached a steady state, you have 200 proteins, okay? And then I work with 200 proteins. Unfortunately, that is often not the reality. What happens is that you get this production in bursts, okay? So if you look at a number of molecules as a function of time, you will see the sharp spikes, okay? So, at some point a lot of proteins were produced and then these proteins slowly degrade and nothing extra is being produced, and then again after some time you get a lot of proteins being produced and so on. Okay. So, proteins and mRNA and proteins in that sense are produced by this mechanism generally called bursting, called transcriptional bursting. Bursting, which means that if you were to tell me that hey, I want to do some calculation that says that is based on this number of proteins, this quantity average number of proteins, since I am writing P, average number of proteins P is perhaps not the best quantity to be working with. And what the best quantity to be working with perhaps is, I should not have written P, I should always try to avoid P, N, N is the number of proteins. So, probably what is rather a better quantity to be working with is the probability to have n proteins at some time t. Okay. So, I switch in some sense from a deterministic framework to something of a stochastic framework, where because of these large fluctuations in numbers, in copy numbers of protein and in copy numbers of RNA, I cannot work with a fixed number, but what I need to work with are distributions, probability distributions. Okay. How many proteins do I expect to have at some particular time? And then you could write down, well, how do these probability distributions in principle evolve with time? And one can do that, but what I'm trying to convince you is that often when you look at biological systems, these systems are often often fluctuating, they are noisy, and these fluctuations are a key part of their function. So, if you always had some base level of protein that was being produced, that is actually not ideal for the cell. Okay. A cell needs to produce particular proteins when it needs it. So, therefore, it has come up with this bursting mechanism, and therefore, the theoretical tools that you will need to study a mechanism like this is this theoretical toolbox of stochastic processes, where you write down probability distributions and then you look at how these probability distributions evolve, okay, and what are the mathematics of dealing with these probability distributions. Depends on your time resolution. Essentially, you can think of them as discrete bursts because it's happening over a very, very short time scale. Compared to, for example, the decay time scale, the production time scale is very small. So, those production events are essentially, you can consider them as discrete events.
Sir, what are the functions of proteins produced?
What do proteins do?
Yes.
Oh, they do everything. I mean, seriously, they do everything. I mean, whatever you think, what does a cell do? I don't know. Anything that a cell does is done through proteins. So, for example, it needs to take in nutrients from outside the cell to inside. It is done through.
Does the rate of production depend upon the requirement by the body?
By a particular cell, yes. Yes. So, there are ways to tune that. So, proteins have, so I don't know how to, so proteins have various functions. Okay. It regulates transport, it regulates anything, whatever the cell does is through proteins. Okay. But I am glad actually now that you asked this question. What it also does is that it regulates when proteins are produced. So, when proteins are produced. So, let us say that I have, here is my genetic sequence, here is where some gene has started. Okay. What was it? ATG, right? GGA something. Okay. So, here is where my gene 1 has started. You get this transcription, translation machinery. Okay. So, how does transcription happen? If you remember this video, what happens is that this big protein called RNA polymerase comes and sits on this DNA, right? It reads off this sequence and produces. So, this is this protein that is called RNA polymerase. It comes and it produces this string of RNA. And then this another big protein, which is the ribosome, that comes and binds to this RNA, and then it produces this protein. Okay. And I should draw this protein. The protein folds, so it has some structure. So, here is this folded protein. Okay. So, this gene was read, it was transcribed into RNA, translated into protein, and now we have this protein.
Can you go backward from RNA to DNA?
No, this is one way. So, DNA to RNA to proteins. Okay. No, so like I said, so this DNA is 10 to the power of 9 base pairs long, right? Out of which only 10 to the power of 7 base pairs actually code for proteins. So, let us say you have started here, somewhere you see a stop codon, okay. So, then gene 1 ends and that produces protein P1. Now, then you have many sequences over here which is non-coding, okay. Then you have another region where gene 2 starts. So, again you have a ATG followed by other sequences. It goes through the same process, it produces a different protein P2. So, all of this information is there in your genetic code. There are different regions which correspond to different genes and each gene produces a particular protein. No, so that is the thing. So, if in principle the entire DNA would have been read at one time, you would have some concentrations of protein 1, some concentration of protein 2 which was roughly constant maybe as a function of time. That is not the case. So, when protein 1 is required, this part is read, RNA polymerase comes and sits here, this part is read into RNA, it produces this protein 1. When protein 2 would have been required, so there is therefore a regulatory aspect. So, that is what I was saying. So, not all proteins are being produced all the time. So, there is a regulation of when a gene is expressed and a protein is produced. No, so the RNA can come, let us say this RNA polymerase protein you mean, this one. So, this is a protein called RNA polymerase, okay. So, it can come and sit here for example, okay. It moves along the DNA, okay. As long as it is not seeing this sequence, it does not start producing the RNA. So, when it starts, that is why you need a start, you need something which tells it when a gene is starting. So, when it sees this sequence, then RNA production starts and that particular sequence is produced until you get to the stop codon, okay. So, that's the now, okay, so is this clear? Okay. What was I saying? So, proteins itself can regulate when proteins are produced. How do you do that? So, let us say this protein P1 has been produced, okay. Now, this protein P1 can come back and let us say it comes and binds to this DNA, right. A protein is whatever, it's some globular object. It has some interactions with DNA in principle. And let us say this protein P1 has come and bound to this DNA over here. Now, when an RNA polymerase RNAP comes and slides on this DNA, it cannot reach here because its way is blocked by this protein, okay. So, therefore, it cannot read this gene and it cannot produce subsequent amounts of this protein. So, what is this? This is a feedback loop. What do feedback loops do? They produce nonlinearities in your governing equations, right? Because the more of protein P1 you have, the less of it wants to produce in this case, or maybe in another case it can be positive feedback, there can be positive feedback, there can be negative feedback, okay? And then you would have to write down appropriate terms in these evolution equations that would depend on the number of proteins themselves, right? So, you would have nonlinearities. These nonlinearities would affect the number distributions, steady state distributions, how these probability distributions evolve with time and so on and so forth. So, I think this is an E. coli gene regulatory network. So, here is one gene, some gene called ARA-C, okay, or rather, yeah. So, here is where this gene starts. These arrows typically indicate the start of a gene. So, here is this gene. So, this gene produces this particular protein ARAC. Okay. What does it do? It goes and binds to 3 regions of the genome. Okay. So, here is my DNA polymer, okay, and here is gene 1 which is ARAC, okay, and that gene is maybe up till here. Okay. Then there is another gene which is in this case a GFP reporter. So, this is GFP. GFP is green fluorescent protein. Okay. And there is another gene which is a LAC-I. So, let us say here is gene 3 and this ends over here, and then here is LAC-I, gene 3 which is LAC-I, that ends over here. Okay. So, I have these 3 regions of my genome. Let us say in this simplified picture, I have this whole string, but there are only these 3 genes. Okay. This region encodes the protein LAC-I, this region encodes the protein ARAC, and this region encodes the protein GFP, the green fluorescent protein GFP. So, when this particular region of the genome is being transcribed and a protein is being produced, so ultimately you get these proteins ARAC, okay, through this transcription, translation and machinery. What this protein can do is that it can bind and regulate the production of all 3 of these proteins. So, it can come and bind here, it can come and bind here, it can come and bind there. Okay. What this diagram tells you is that this arrow sort of a symbol, what it means is that this is a positive feedback, which means the more of this protein you produce, the more of GFP it wants to produce, the more of LAC-I it wants to produce, the more of itself it wants to produce. Okay. On the other hand, this protein LAC-I is a repressor, which means the more of LAC-I you produce, you see this sort of a arrow, I do not know what you call it, whatever, this flathead arrow. So, that means that this is a repressor. So, the more of this particular protein you produce, it wants to repress its own production, the production of this, the production of that. Why a GFP over here? The GFP over here is in some sense a reporter. So, you cannot look inside a cell and say that, well, this particular protein is being produced in some sense. You have to have some way of estimating whether a protein is being produced or not, and that is done through this fluorescent protein. So, essentially this encodes a protein which fluoresces. In this particular case, it fluoresces green. So, that is why it is called the green fluorescent protein. People have various reporters in biology, red and so on, yellow and so on. So, what that means is that when this protein is produced, you will see spots of fluorescence which are green in color. And then if you look at single cells, okay, so this is a single cell time. So, there are, I do not know how many, so 1, 2, 3, 4, some 20 cells or so. As a function of time, you are looking at the fluorescent intensity of this green protein as a function of time. Okay. And what do you see? No green protein, very high green protein over here in this region, and then again no green protein, again very high green protein, again no green protein, and so on. Okay, on off, no steady fluorescent level. So, nothing like this, something like this. Right. In another cell, and again these are not coded, these are not not in all cells, they are not happening at the same time. In another cell, for example, there is this burst over here and then something over there and so on. So, you get this random pattern which tells you that the underlying dynamics of these gene regulatory networks, these are called gene regulatory networks, these underlying dynamics of these gene regulatory networks are stochastic. Okay. There is a lot of fluctuations going on, and ultimately these determine when a critical concentration of this fluorescent protein is being produced, so that you can see that. So, the whole of this lecture is sort of aimed at convincing you that these quantities inside cells are fluctuating quantities, and what you should be studying are probability distributions and the evolutions of these probability distributions. Okay. There are, of course, and because of these nonlinearities, because of these feedback loops, you can get very complex dynamics, and these itself are interesting to study. If you put many of these cells in contact, you can get further interesting physical phenomenon. For example, here are many cells in contact, and initially you will see that each cell was fluorescing at random times, but ultimately after some time has passed, some 150 minutes or so, you start getting synchronization. So, all cells which are talking to one another start fluorescing at the same time. So, you get these bands. When things are fluorescing, everything is fluorescing together. When things are not fluorescing, nothing is fluorescing. Okay. So, these are emergent collective behaviors. Okay. So, this is, if I had just told you that, hey, look, here are these 2 genes, ARAC and LAC-I, and this is how they behave. A priori, I would not have been able to tell you what signal you would see if you had put hundreds of these cells together. Okay. So, in that sense, this is an emergent collective behavior, and again these sort of emergent collective properties are interesting to study from the point of view of physics, where you know the microscopic rules. The biologists or a chemist, a biochemist has come and told you what these proteins do to one another. So you know the underlying regulatory network. What you want to understand are what are the macroscopic consequences of that network. And this sort of framework of stochastic processes, of probability distributions and so on is what allows us to do that. So just to give you an idea, so when I say that there are so many proteins in the body, so these proteins have a huge range of sizes as well. So for example, if you think of the smallest human protein, which is insulin, that's only 54 amino acids, and this is this structure of the insulin. The largest human protein is titin, which is 34,000 amino acids, and that is the protein in your muscles. So this is what allows your muscles to expand and contract. You have some distribution, again, if you think about protein lengths in amino acids, you have some distribution. Roughly around 300, 400 is about where the mean is. So the average length of human proteins is around 485 amino acids, and the average mass is about 53 kilodaltons, 1 dalton is something like this. So there is a broad variability in sizes. But more than that, there is also for even a particular protein, there is a broad variability of in that particular protein number or concentration as the cell goes through the cell cycle. Okay. So this sort of variability is what we want to focus on in this course, and we want to ask that by looking at these sort of variabilities systematically, what sort of physical insights into organization of biological systems can I gain? As a physicist, what are some of the underlying principles that I can try to understand by using these tools of probability and all of this machinery over here. Okay. So that's sort of the aim of this course. So that's what we'll try to do. Huh? Oh, textbooks. Thank you. I have a slide at the end. All right. So this, as non-biologists, I feel this book by Rob Phillips is a nice introduction to biological systems. It's not the book that I'll be following for this course as such. Okay? It's just a book that tells you a lot about, the biology and how to think about biological systems as a physicist in general. So if you're a non-biologist, this is a very good book to sort of gain an idea of these various biological terms, DNA, RNA, RNA polymerase, this protein, that protein, transcription, translation, so on and so forth. So in that sense, it's a good background book to be reading. The main three sort of textbooks from where I will do the theory, the actual pen and paper sort of theory, are these three: stochastic processes in cell biology, stochastic processes in physics and chemistry, Van Kampen, and perhaps certain sections from Gardner, Handbook of Stochastic Methods. So this Van Kampen and Gardner are the standard physics textbooks for physics of stochastic processes. So these two have nothing to do with biology. These are pure physics textbooks. This is in some sense these stochastic processes applied to biology. So I'll take some examples from this book as and when I come across them. The remaining ones are, again, perhaps not particularly to do with the course. These are simply a set of good biological physics books if you're interested in biological physics as a subject from different perspectives. So Phil Nelson has these two books about biological physics. Bialek has this very nice book about what are the overarching principles, physical principles behind biological organization. So it's again, this is not a textbook, but it's a good book to sort of be reading if you're interested in this subject. Helmut Schissel has this book, Biophysics for Beginners, and this is on the other hand different sort of book which deals with some specific biological systems in a lot of mathematical detail. So Mathematical Biology 1 and 2. But again, like I said, these are not books that I will be particularly, I might take one or two things from here and there, depends. As far as books go, probably these two are the main books, Van Kampen and Breslov. Having said that, I mean, that is for the theory aspect. A lot of the examples that we will be working out are taken from all over in some. So the disadvantage of a course like this is that there is not one very good textbook that I will follow, and therefore, not one very good textbook that I can tell you for this. So, but as and when I teach something or I discuss something, if I'm taking it from a paper, I'll tell you what paper I've taken it from. If I've taken it from one of these books, I'll tell you which book I've taken it from so that you can go back and look. Okay. That's the idea.